Thank you so much for stopping by my poster. I thought I'd do a quick recording to hopefully help you through some content. If you're here for the quick version of the PowerPoint or of the poster, this slide sums up some of the main findings of our work. Some of what we've done over the last year and a half is built out a semi-supervised pipeline to annotate all of the molecular targets in SARS-CoV-2. This is to identify the genes, proteins, and functional domains, and do so in a way that is without the requirement of a single reference genome. The pipeline itself was benchmarked significantly for accuracy across multiple different facets. And on the bottom left-hand side is a schematic of the SARS-CoV-2 proteome. For protein and domain sequences, we have a 98.5% and 99.1% set membership accuracy in those two data types. And that means of the different molecular targets that should be identified in the SARS-CoV-2 genome, we were able to identify with that percentage of accuracy. In addition to that, we're able to have high sequence identity for PP1AB and other key proteins like spike glycoprotein and domain sequences, including the receptor binding domain. So not only are we able to identify all of the correct targets, but those sequences are based in biological reality. Beyond this, we characterize the variants that emerged of the spike glycoprotein, and in this case, we were working with a corpus that was collected earlier in the pandemic, with genomes dating up until fall of last year. Using the semi-supervised method, we were able to predict sequences that accurately identified the D614G mutation. And as you can see on the top right-hand side, the orange line is that variant overtaking the ancestral lineage, which is shown in the green. Beyond this, we characterize the different variants as they relate to exposure region and represent those at the bottom right. As far as the different variants that emerged, not only did we identify the D614G mutation, but in a small number of proteins, we identify the N501Y mutation, which is one of the causative mutations involved in some of the most dominant variants that we're observing in the pandemic today. This has all been published under, it's at a preprint for bioRxiv. The link is down below, and it's under review currently. So if you have any questions, you can refer to the paper for a bit more. And if you're interested in hearing some more details, feel free to stick around and I will keep describing some of the methods. Awesome. So if you're interested in hearing a little bit more about the method, I'll of course start by giving you a couple of resources that you can use in your research. So related to the semi-supervised method to identify SARS-CoV-2 molecular targets, we've released all of the code in our public GitHub, the links included here, and put this together as a Docker container where you can pull the Docker container, build it, and then run the execution to on any SARS-CoV-2 genome. This has been tested for the paper with a corpus of over 66,000 genomes, and then we've continually annotated and updated genomic and molecular target information as the pandemic is continuing. And so this has been applied to hundreds of thousands of SARS-CoV-2 genomes at this point. As it relates to the repository, we've included annotation with just the Wuhan reference strain, and so that's some example data for you to try it out. And feel free to take a look at the GitHub, and then the link to the bioRxiv is included there. So as it relates to genome annotation for SARS-CoV-2, we wanted to have a couple different considerations for what good genome annotation looks like. And for the most part, typically that starts with making sure to identify good data coming into the pipeline. And so one of the things that we did in this work is to actually benchmark what are the quality criteria that we should recommend as it relates to working with SARS-CoV-2 genomes. From the genome annotation work, you have the ultimate goal of minimizing truncations of gene and protein sequences. We want to yield gene and protein sequences that are comparable to known references and maximize overall gene and protein annotations for a genome, making sure that you're identifying all of the correct targets that should be present. On the right-hand side, we've included the different criteria that we evaluated for the efficacy of identification of these targets. We benchmarked a couple commonly used criteria, and the one that's shown here on the screen now is what we find to be the most accurate. Some of the other criteria that are in play in some of these different analytic platforms actually ended up being a little bit too stringent and would inadvertently limit data. And so this is the recommendations that we've seen, and the benchmarking is included in the paper. Um, One point I thought I'd add in here is some work that Akshay Agarwal in our group led around quantifying the genome quality in these different public repositories. And so often in bioinformatics, the adage of garbage in, garbage out is so completely in play. And there's always the reminder that, really not all public data is equal, and you should trust no one. But, with jokes aside, we quantified the amount of unknown bases or ends in a given genome, and then also the degenerate bases that are observed in a given genome. And those are quantified here on the two different data sources that we worked with for this paper, in GenBank and in GSED. And then the dashed line indicates the quality thresholds that we applied to this corpus, where genome dots that are, points that are to the right of the dashed line are no longer included. Of the genomes that were below our quality threshold, 10% of those, or 10% of all GSED genomes, were below that quality criteria. And for GenBank genomes, about 3% were below that quality criteria. In addition to that, as part of our general processing for the functional genomics platform, we identify when genomic information has been exactly seen before. So by creating a MD5 hash of the genome sequence itself, not including the header, just the sequence, we track duplicate or replicated genome sequences. And in this case here, we wanted to quantify, above the horizontal line, the data from the different sources, GSED, GenBank, and of course the RefSeq single reference genome, the Wuhan reference strain, and then the occurrences that we see within a repository and between repositories. And so that shows the genome accessions and genome hashes that are replicated between those. And you can see, within, GSED to GSED, GSED to GenBank, GenBank to GenBank, and so forth, to quantify the duplicated genome information. Some of this can be an actual data artifact, and some of what we did is to quantify that by metadata overlays or similarities. But then at the same time, too, because of the nature of the pandemic and the high volume of sequencing of a single organism that's going on at this current time, some of this is just going to be oversampling of an organism that is only evolving at a certain rate. In this plot, we are demonstrating that the application of this quality criteria allows for the better identification of protein targets within the SARS-CoV-2 genomes. So this violin plot shows genomes in the salmon color that are above our criteria, that are active, and in blue are below our criteria, are marked as inactive. In the middle is what the expected protein sequence length is. And using a KS test, we identified that the active genomes, the genomes that we apply above our criteria, have a length distribution of protein length that is more similar to expected than the inactive is. And you can see too that overall we yielded less truncated proteins in our active genomes, and this actually yielded an observed over expected protein length of 99.1% across all proteins. In the blue, you can see that the volume to the bottom and then also some of the blebbing in that 2,000 to 4,000 range are all indicative of those truncated proteins. And then at the top, which is actually for replicase polyprotein 1AB, the volume there is not as wide as what you observe in the expected distribution. So now thinking about the genome annotation work, so we had to implement a few different things to improve upon the annotation method that we'd been previously working with. So as it relates to the functional genomics platform, we'd worked in bacterial genome analysis for several years. When COVID hit, we had this structured repository and analytic pipeline that is suited for prokaryotic annotation that we thought we could, readily adapt. And the reality is the sequence had never been, SARS-CoV-2 has never been seen before, and some of the off-the-shelf capabilities weren't well suited for that. So we had an initial implementation and then realized that we needed to do more to be able to improve the accuracy and robustness of this method. And some of those different changes include better control for processing of short genomes, where PROCA was previously working in metagenome mode because the size of SARS-CoV-2 is so much smaller than what it's expecting. So we force a single genome mode instead of metagenome mode. And then, of course, too, bringing in the latest SARS-CoV-2 UniPro reference data to keep pace with the pandemic discoveries. And because of that, when we are constructing our references, we it intentionally increased the leniency of the reference data that's built, in part because in this nascent state of reference data for SARS-CoV-2, and this work really started about a year ago, well, over a year ago now. We wanted to include putative proteins, things that hadn't necessarily been experimentally confirmed, because the data was coming out so fast as far as reference data availability. So we wanted to make sure that those were incorporated as well. And then beyond that too, as it relates to our domain annotation pipeline, we've brought in the special release that UniPro provided to create and identify the domain signatures. Since we initially did this work, we're now on version 85, which has a huge expanse in domain to KEGG mapping, so that each of these proteins domains can all map into the pathways that they're associated with. As it relates to identification of the different proteins for SARS-CoV-2, one metric that we are observing here is the set membership accuracy. And what that means is when you consider a SARS-CoV-2 proteome, you want to identify all of the known proteins that can be present, or that should be present in that genome. And in this case, you can see on the left-hand side, on the bottom plot, for each of those proteins, the dashed line indicates the total number of genomes that were annotated that were above our quality threshold. And you can see on the majority, each of those proteins are identified with very high set membership accuracy. And across all proteins, it's an average of 98.5% set membership accuracy to identify all of those different proteins. And then beyond that too, we can look at which of these different proteins have the most number of unique sequences that are present. And you can see here, spike and nucleoprotein have the highest number of mutations when you're considering the structural proteins. And this is, of course, what we've observed over the last, several months as these different variants occur. Many of those mutations are occurring in spike and the nucleoprotein. But also replicase polyprotein 1A and 1AB have a higher amount of distinct sequences or non-redundant sequences, which can be a little bit of a product of the length of those proteins themselves, of those polyproteins. Beyond this, we wanted to make sure that the identification of our protein sequences were based in biological reality. And one of the challenges for autonomous viral annotation for SARS-CoV-2 is related to this transcriptional slippage site that occurs in the replicase polyprotein 1AB in between ORF1A and ORF1B, where here we had to intentionally add into our methods to be able to overcome the split products that can occur at this site. So off the shelf, PRACA was not able to produce a single full-length protein product in any SARS-CoV-2 genome. And so Gowrie Nayar, one of my teammates here at IBM Research, she built an algorithm to be able to identify this slippery site signature with robustness, and then bridge the gap between these two different regions of the gene and protein products, and then apply the appropriate program -1 ribosomal frame shift to create the correct protein product. On the right-hand side is the plot that demonstrates the sequence identity to references, and it seems like it's a pretty vanilla plot, so to speak, because it's a bunch of white space in the middle, but it's actually a really good thing because on the y-axis is the percent identity to the PP1AB reference sequence, and you can see that over 95% of we have over 95% of our sequence identity and 99.15% of our protein sequences. And the frequency of those different sequences are indicated in color and then also in that point size. So the majority of our sequences have very high fidelity to the references and are able to achieve that full-length protein product that is going to be have higher accuracy to the reference. In our protein sequence analysis, in our protein domain sequence analysis, we also wanted to investigate set membership accuracy and then also the sequence fidelity to known references. In this case, we took a targeted look at spike glycoprotein for the domains that are known to be involved in that, especially because spike glycoprotein is one of the key gatekeepers for host cell invasion, the target of all of our vaccines, even many tests, right? So on the lower left-hand side is a plot indicating the set membership accuracy for each of the domains that are shown on the top right, which is a screen grab from Unipro. For the two domains that are very, very lowly present, those are actually false positives, and they're indicated at a very, very low amount. I'm included in here for data completeness. And then the point for the IPR 043473 that's occurs doubly to the set of spike sequences that are being analyzed here. And that actually is the domain that's in that pink color on the bottom, that's actually just split into two locations. And so that's why the total count of sequences for that domain is overrepresented for the number of spike sequences, but it's appropriate as such because the domain is located into two fragments. On the right-hand side, we have demonstrated, or we're indicating the sequence similarity to known references here. Again, showing you percent identity as a box and whisker plot per domain. For those three domains, I'm sorry, that were false positives, we compute those. We indicate that the percent identity is -1, meaning non-applicable, since there's not actually a reference in that sequence. For those domain sequences. But of the other domains, we're able to have greater than 98% median sequence identity. And then for the set membership, 94.4% set membership accuracy on average across the different domains as well. As with so many bioinformatics methods, it's important to demonstrate novelty and an improvement in the field for what has actually been contributed here. And this is actually benchmarking work that Akshay Agrawal led, where we did a direct comparison on a set of 100 different randomly selected genomes. And here we have the observed over expected protein length across three different tools, our method through the functional genomics platform, VAPID, which is a viral annotation tool, and then PROCA with your off-the-shelf usage. If a protein is missing from any of these analytic tools, it's indicated as 0, and so you can see that the functional genomics platform has less truncations overall and less missing proteins. And between our method and the other two, we achieve a 6.4 and a 1.8 fold increase in protein annotations over base PROCA and VAPID. Beyond this, we wanted to say, okay, great, so what can this data actually tell us about SARS-CoV-2 and about its role in our public health and the evolution that it's undergoing? So here are a couple examples of phylogeny demonstrating the different lineages and the nomenclature that's proposed for SARS-CoV-2, right? Is, it's loosely quantified that the virus on any given transmission chain can have 2 mutations per month. And then, as you know, in December really is when we started to see a bit more of an expansion of the different variants and variants of concern. And this was taken, this screen grab was taken a few weeks ago when the B117 variant indicated in that dark purple on the bottom right-hand side of the phylogram is really expanding. And you can see its indication as a dominant proportion of all of the different pie charts across the globe as well. And so what we looked at in the annotation of the corpus that we used in benchmarking this method is that we were able to identify the D614G mutation accurately and also better quantify the timeline to which it overtook the ancestral sequence. Beyond this, we were able to look into this corpus and identify the N501Y mutation. And this corpus was obtained just prior to September of last year. And in some of these early sequences, we see this mutation occurring in Oceania, North America, in late summer. And this mutation is now, it's in the literature, it's been observed to have multiple introduction events, and it now leads the dominant B117, B1351, B1617, .1 and .2 variants, which is, Delta variant and so forth. And then in addition to that, we quantified the variant present across different exposure regions. And you can see in that third up from the bottom sequence, the D614 variant that is much more prevalent across different regions. And then you can see a speckling of other variants that may be observed across the world and maybe a little bit more prevalent in some of the other regions. And this is the characterization of, in this case, just spike glycoprotein variants as well. With that, I will thank you so much for your attention. This is the follow-up of the TLDR from the beginning of the poster presentation. I hope that we've convinced you that we've put together a method for accurate large-scale genome annotation with high accuracy and set membership and biological sequence identity, and that this data can be useful in your identification of important variants and how that may reflect and interact with different vaccines, diagnostic tests, and so forth. And with that, feel free to take a look at our platform, ibm.biz/functional-genomics, and then feel free to read our preprint further and reach out to me if you have any other questions. Thank you so much.